Alright, so today we're going to be learning about meiosis and crossing over. Um, I'm going to want you to know about how meiosis leads to genetic variation. That's why we're studying it. How does it lead to genetic variation? So make sure you have your title, meiosis, and you have your target written on this. You're setting up your notes for Cornell Notes. If you need time to do that, go ahead and set that up now. Push pause. Alright. Alright, so one of the first words we're going to learn about is somatic cells. They're your body cells. And one thing you know about them is if we're talking about people, humans, we have 46 chromosomes or 23 sets of chromosomes. And you're looking at a set of chromosomes in the picture right there. Right there you have what's called a homologous pair. That's those two X-looking things. You have a set from your mom and one set from your dad. If you look at that X, you have one half right there. One half, that's a chromatid. The matching pairs there, that's what makes a chromosome. So two chromatids make a chromosome, and if you have the same ones that code for the same sorts of things, like hair color or height or I don't know, whatever, it's going to be two of the same pairs of chromatids. That's going to give you your homologous pair. So we would have 23 homologous pairs for humans. So here again, we are looking at a karyotype. A karyotype is when you have all your chromosomes mapped out. This is an illustration, and they're sorted by length, so how big they are, by where their centromere is, and the centromere is that middle part where they connect, and then gene location. So that's what we're showing with the little banding here. What genes are where. Autosomes are what we call the chromosomes that code for your normal characteristics, non-sex related characteristics. And you have 22 of them. So in this karyotype, you see again they're sorted by size and where their centromere is, where the bands are. They're all organized 1 through 22. They're paired up. And then this 23rd pair is a little bit different. You notice that one says X and that one says Y. On humans, the sex chromosomes are the X and Y, the 23rd pair here. And what's possible is you can have a different size and different genetic composition, whereas in the autosomes, they're all the same sorts of stuff. So here we have X and Y. This is a boy. The XY means boy. The Y can come only from the dad. The X comes from mom or dad. So you always have a 50% chance of having a daughter or a son. And that's going to be determined by what the dad does. All right, now take a minute and I want you to try and answer this question. If there's one pair of sex chromosomes, how many pairs of autosomes are found in humans? You can go back, look at the video. If there is one pair of sex chromosomes, how many pairs of autosomes are found in humans? All right, you should have paused it, giving yourself a chance to find out the answer. If you haven't, pause it now. I'm about to give you your answer. Okay, there are 22 pairs of autosomes found in humans. Alright, some more fun and always exciting vocabulary. Diploid cells. Alright, diploid cells have two homologous sets of chromosomes. So that's like most of your regular body cells, your somatic cells. They all have two sets of chromosomes. So like cells in your finger or heart tissue, whatever, it's going to have 46 chromosomes. 2N. Right? Haploid kind of sounds like half. It's 1N instead of 2N. And it has one set of chromosomes. So the only things that are haploid cells in people and animals are going to be your sex cells. things like sperms or egg. So that is what are some examples of haploid cells. And what we're trying to do in meiosis is we're trying to take diploid cells and turn them into haploid cells. Alright, so what I want you to do now, a little review, go back, look at your mitosis notes, and look at your pictures you've drawn. That's my phone, sorry. What are the phases of the cell cycle? Go back and try and look for that. Give you a hint, start off with like G1. Alright, hopefully you push pause again. It goes G1, S phase, G2, and then you have mitosis and cytokinesis. And again, G1, the cell grows. S phase, it gets ready for replication, makes copies of the DNA, makes copies of other organelles. G2 happens, again, it's getting bigger, it's growing, making copies of organelles. And then mitosis happens and it splits. Meiosis, we're going to see, is a little bit different than mitosis. We are going to make gametes, and if you notice, gametes are different shapes. You have the egg, you have the sperm, and as I mentioned earlier, gametes are haploid instead of diploid. So in mitosis, we divided once. In meiosis, we're going to divide twice. Meiosis happens in the gonads. So in humans, that would be an example, the testes or the ovaries. But plants also do meiosis, and this would happen in different parts of the flowers. And the idea here is we need to have two haploid cells to come together and make a zygote. So fertilization is what we call when this happens. You have a sperm, comes together with an egg, and you get a zygote. So a zygote is just that one cell, and it's going to later on turn into more and more cells through mitosis. But again, fertilization, we have a sperm and an egg come together and then makes our zygote. Here's kind of another picture of the same thing. We have female and male, they both go through meiosis. Guy makes the sperm, female makes the egg. Those come together through fertilization. They become 2N, they differentiate and grow, and become humans. Cycle goes over and over and over again. Alright, this process should look pretty familiar. It looks a lot like mitosis, but there's a couple little differences. One difference that's really important here is something called crossover. If you notice, the chromosomes are really close together and they're switching their little parts. That's crossing over, and that's going to add even more genetic variation. It's going to add more changes. Also, right here, you notice they're lined up side by side. In mitosis, they're all right on top of each other because each cell's getting one copy. Here, they're lined up in tetrads or homologous pairs. Here is meiosis 2. So if you notice, it has two phases. These two cells come from the last picture, and each of them is going to end up splitting. All right, again, we have sometimes more crossover. They are moving along, and we're turning into 1N. So here we're 2N, here we're going to 1N. So we started off with one cell in meiosis 1, and at the end of meiosis 1 we have two cells. Meiosis 2, we start with two cells, and we go into 4 separate cells. Alright, so here's a little video showing meiosis happen.
Meiosis, just as it does for mitosis. But as the DNA condenses, each chromosome finds its homologous counterpart. These matching pairs of chromosomes hold equivalent genetic information, one set from mother and one from father. Next comes the first shuffle of nature's genetic deck. The two homologs trade genes in a process called crossing over. This forms new hybrid chromosomes. The spindles then place maternal and paternal chromosomes randomly on each side of midline. It is another shuffle that can lead to a host of different genetic outcomes. Finally, spindle fibers pull the homologs apart. This leaves the sister chromatids intact. With twice the amount of genetic information the cells will need, nature must cut the genetic deck in two. This second division produces 4 unique sets of chromosomes. Because they carry half the genetic information, we call these haploid cells. Many of them will mature into sperm and eggs, the reproductive cells called gametes.
Alright, so take a little while, look at this picture. Go ahead and push pause when you're ready and tell me what are 5 differences you notice between mitosis and meiosis. There's probably lots more than that, but take a look at 2 pictures, look back at your notes and come up with 5 differences. Alright, so you've paused it for a little while. I'll tell you 5 that I can see really quick. 1, mitosis doesn't have any crossover. 2, if you notice, in metaphase 1 here, the homologs line up in tetrads, whereas here they just line up all on top of each other. So that's one difference. And then again, that has to do with the crossover as well. Another difference, mitosis ends in diploid cells, where mitosis ends in diploid, where meiosis ends in haploid. Let's see, what else? Also, meiosis has 2 cell divisions, where mitosis has 1. That's all I can think of right now. So I've mentioned crossing over a few times, and here's a little nice, easy picture, and a couple of the vocab words you'll need to know. We have crossover. Here are my 2 homologs, my homologous chromosomes, and what is called where they cross over is called the chiasma. And here you see the different colors represent the different chromosomes, and they've switched. So if we're talking about hair color, maybe this one was coding for red hair and this one was coding for brown hair. And here we switched it over, and now that means that this one has some of those brown hair color characteristics, and this one has some of that red hair characteristics. So they've changed the DNA sequence just in a little bit. So what crossover does is it's adding to changing our genetic makeup. Because one of these came from mom, one of these came from dad, we're kind of mixing it up a little bit, making things more different. Now, go back, take a look. Name 2 ways in which meiosis increases genetic variation. Go ahead and push pause. Alright, so genetic variation changes because, 1, we have fertilization, where we have 2 different sets of DNA from dad, DNA from mom. It gets reshuffled around. And then another way would be through that crossing over, which makes those sperms and eggs even more different. Okay, now you're going to have to do a little work. Identify the phase. This is meiosis, not mitosis, so this is meiosis. Go back and look at your pictures. I know you drew some. What phase is this? Okay, I'll give you some hints. 1, if you notice, they're not lined up in tetrads, and we just have a single chromatid. So we are pulling apart, and we're going to be making 4 cells. So that's another hint. If you still haven't figured it out, pause it, look in your book, see if you can figure out. So what is this down here? That's showing some crossover. Alright, the answer is this is anaphase 2. Our cells are splitting apart. Sorry, correction, not our cells are splitting apart, but the chromosomes are being pulled apart to either side. How many chromosomes would a cell have at the very beginning of meiosis? That's a good question. We're talking about people, not like a strawberry. But how many chromosomes would a cell have at the very beginning of meiosis? Alright, so this one is a little tricky. Um, you probably said 46, and that's a really good guess because your normal cells have 46 chromosomes. But we need to double that because we're going to be splitting. So remember, in S phase, we replicate our DNA. So it would be a total of 92 chromosomes before meiosis happens. That's going to split into two so that we have two cells that are diploid. That would be 46. And then we would have those split in half again, and we'd have four daughter haploid cells that each had 23. So we go 46. We go 92 to 46 to 23. That's two cell divisions there. All right, that's it for now. Go ahead. You're going to need to make sure you know all the phases. You're going to want to memorize those. Memorize what happens in each of those phases. But go ahead and write your summary, write your questions. Take a few minutes to do that. And then go ahead and put this on the appropriate page in your interactive notebook. Thank you very much. Thank you for doing your homework.